Hi guys, welcome.
If you struggle with focus, this sound might change everything.
Hi, how are we? Put you over there. I'm hoping that you can hear me good. Right, okay, so I know TikTok can hear me. I'm hoping, yes. How are you? I've just started immunity today. Nice. Okay, and you can hear me. Tails, can you hear me on YouTube? Had a nightmare earlier. Perfect. I did a GCSE live earlier and I must have been halfway into it, 30 minutes. And then somebody popped on to YouTube and wrote a comment and said, "We can't hear you, there's no sound." I was like, no. Do you want to cover B cells and T cells? Sure. No problem there whatsoever. It's a funny one. It's one that comes up so continuously that students struggle. No, this isn't GCSE. This one is A level now. GCSE was my one I did at 6:30 earlier, and I'm having to download the video, adapt it, and then I'll re-upload it into YouTube because I just kept going. So I'll edit it slightly. Chills, you're AQA, aren't you? If I'm right in what I'm thinking, I'm sure you're AQA. Okay, so I feel like the dreaded voice issues are going to be coming this next couple of weeks. Okay, so if we're going to look at the T cells and B cells, the first thing I would say to do when you're looking at anything to do with immunity is, please can you go through neuronal communication for OCR, please? I have a test tomorrow. Yes, I will. Let me just quickly go over T and B cells, but Ava, whilst I'm doing this, if you want specific stuff for neuronal communication, have you checked my 5.1.3? Hold on, I might be wrong on that one. Hold on. What is it? Neuronal communication is 5. Oh, I was right. 5.1.3. My 5.1.3 playlist has all the content there, and then if you want to have a quick view of that and then pop back and ask a question, if you're not sure, that could be something that might help you specifically if you've got a test tomorrow. But if not, if you've got like actual questions, specific question, please ask away, and then I'll add in some more. So just to go through quickly, Tills, you've been learning about the B's and T cells. So you have different types. If we were to go lymphocytes and break them down, I'll do it very basic here. Hannah, it's not GCSE, we're into A level now. We've got our B lymphocytes and then our T lymphocytes. You are very welcome, Eva. So you've got your B lymphocytes and your T lymphocytes. And then if we split our B lymphocytes down, we would split them into plasma cells and memory cells. And then we would add in the plasma cell is the one that makes the antibodies. And if we're going to go T cells, we would then split that one down into our helper cells. What I was doing there, by the way, when you're watching me, is I was actually closing my eyes to visualize, because I'm like, I know that you've got different exam boards, and so I'm like, right, which one am I doing? So I would say the killer cells, but because Tills, you are the AQA, it's cyto... Oh my gosh, my brain's just totally switched off. Cytotoxic. Wow, that did not work there for a second. Cytotoxic, whereas OCR, we just like to call it the killer cells, and then the memory cells. But you won't get marked down for saying hi Jazz, you won't get marked down for saying killer cells or cytotoxic cells, but it is important when you're looking at it that you go specifically on the stuff that your exam board have decided as that's what they want you to do. I don't know if you remember me from last week. I was the one who had the rest for respiration. The test on respiration. Oh, I'm glad it went okay. Oh, I'm really glad it went okay for you. Cytotoxic. Oh, you're very welcome. You are very welcome. 1 second. I'm just putting something up. So if you were looking at, and this is what I would say, and this is what I do with my students all the time. So I'm just going to quickly stuff that in there. Whenever you are doing your revision, always refer back to your specification. So Tails, just to show you here, cytotoxic T cells is what you need to know. Whereas if I go to the OCR one, oh, I'm going to so lose my voice, I can tell it already. T cells. Where are you? So then if I go to OCR's one instead, and they bring that one in, and this is why I always advise you guys should have your specifications open as you are doing the revision. So let me just take that off of there. Oh, I did not mean to do that. Crumbs, I'm going to have to copy again in a second. So whenever you're doing this, this is one of the key things that I would say to you, that you are comparing. A lot of you say to me, what's the difference between OCR and AQA? This is a prime example. So OCR, this is part of their specification, and they have named it as being the T killer cells, whereas for the AQA, you need to know it as cytotoxic cells, TC cells. So always double check what it is that your exam board wants you to talk about. So you can see as in cell signaling, they've said interleukins, whereas on this one, they haven't actually specified what it is that they want you to use as the description. So you can call it cytokines, you can call it interleukins, it doesn't really matter. So lymphocytes, you've got your B, you've got your T cells, you've got your plasma cell, you've got your antibodies, your memory cells, your helper cells, excuse me, killer cells or cytotoxic if you are AQA. And this is why a lot of the time a student say to me, oh, but I'm doing this exam board, will your revision guides be good for me? I haven't actually done one on this topic here, I will at some point. But there is so much overlap, it's just about that little bit of specificity that you have to look at what your exam board wants you to use, because that's where, when people say, oh, the mark schemes are so tough, they're strict because that's the kind of answers they're looking for, because they specifically put it into the specification. And if you're watching different exam board videos, or a video that says, I'm OCR and AQA and EdXL and CIE, yes, the science generically is the same. However, the onus is on you, not the person who's making this, to say you should be checking against your specifications. So just do make sure that you are going through that. Ava, are you still in here with us? Tils, did you have any other questions on what you've been learning about? I'm assuming you're interested when we talk about the chemicals that are used. the chemicals that are used, so the interleukins, or your one, hold on. They didn't specify, did they? Yeah, they just said it stimulates. So this would be interleukins, or you could use the term cytokines. And they are going to stimulate those B cells. They are going to stimulate the T cells. They are going to stimulate phagocytes as well. So coming over here to our phagocytes, and the phagocytes that are going to do phagocytosis. Miss, this is kind of related, but do we need to know about perforin? So perforin would be the chemical that is produced by your cytotoxic cells. So perforin is an example of the chemical. They may give you a question that talks about how it's hydrogen peroxide and not perforin. So it's basically understanding that the killer T cells, the cytotoxic cells, are going to produce chemicals that are going to destroy the cell. So that's the important thing, is that it's either hydrogen peroxide or it's perforin that's going to be used to destroy the cell. So if they want to talk about like hydrogen peroxide and understanding that that would affect the membrane permeability, or the perforin would affect the membrane, making it porous, actually breaking apart the cells, they don't go into that much detail. So the key thing is either naming it or them giving you some information, you describing what it would do. Would the pathogens then engulf any debris? Be careful, be careful. So the phagocyte, a pathogen, so what you just said there was the pathogen. The pathogen is the microorganism that causes an infectious disease. The phagocyte is what does the engulfing and digesting, and it engulfs and digests any of the pathogens, the antibodies, the debris. And when you talk about apoptosis, when you're talking about programmed cell death, that is, well, yes, the phagocytes engulf the apoptotic bodies, and when they engulf the apoptotic bodies, they can then break them down. So it's a way of recycling and the organelles that were no longer useful in the cells that were destroyed. But apoptosis is programmed cell death. That's a different scenario to, yeah, I know, it's a different scenario to the phagocytosis that's occurring. So just, yeah, there are so many words, and that's one of the things that you really have to work your way through as you're doing it, because you don't want to get these words confused. So you're absolutely right. Antibody, antigen, antibiotic, for example, of some of the ones that you've written down. These all have those same starting point, and if you get it wrong, you will lose the marking points. So it's really important that you know the difference between the antibody, which is going to be the thing that we are going to use to bind to the pathogen. The antigen is the surface tag for foreign cells. And then if we're looking at the antihistamines, yeah, absolutely. Or if we talk about antiparallel, and there's so many different things we can throw in here. Antibiotics are the chemicals that stop bacterial metabolism / destroy brain, etc. But there's so many. It's like when you start doing hormonal communication and you start doing the gluconeogenesis, the glycogenolysis, the glycolysis, the so many different ways to go. It's really important you know the difference between those words and you slow yourself down. Are antigen presenting cells phagocytes? Yes, they can become antigen presenting cells. If a cell is presenting the antigen on its surface, it is then an antigen presenting cell. So a macrophage is a type of phagocyte that would become an antigen presenting cell if it has encountered a pathogen, engulfed it, digested it, and then displayed the antigen on its surface. Yes. Ria, you know in meiosis 1 at the end of telophase, is it classed as diploid or is it haploid? No, it's haploid. So that's a really interesting one, Ria, that you've just said. Hold on, let me see. I'm in the process of building my new workbook on patterns of inheritance, and I have been doing some new diagrams. I think one of them might be useful in this. Give me a second till I find it. Here, let me take this one. Okay, so here is my new diagrams. Okay, so we have got meiosis 1. So this first part here is meiosis 1. This one here is meiosis 2. So meiosis 1, you know that what's going to happen during anaphase is that the homologous chromosomes are separated. When you look at meiosis 2, this is where the sister chromatids are separated. So you've got meiosis 1, anaphase 1, homologous chromosomes are separated. So right now, if we say that the blue is 1, 2 is the red. So in this cell we have 2N, we have a diploid set. We have 2 lots of chromosome A and chromosome B. Whereas when we get down after the first meiotic division, we have only 1A and 1B. We have only 1A and 1B, so it's already haploid. So does that make sense? Now, it's an interesting one of the question as well that you've asked, in the fact that you've said at the end of telophase, because the end of telophase, the nuclear envelope has reformed, you would therefore say that it is haploid, but because the cell hasn't yet completely divided by cytokinesis, that's where it's a bit of a sticky place where the specific bit of which you've named, but we would say the nucleus is a haploid nucleus. So it's really important that you're using and thinking about that terminology rather than it's the cell is haploid, it's the nucleus that's haploid, specifically at that point. Miss, I learned a really cool fact. When we have fetuses, we have many cells containing antigens and our lymphocytes attack our cells, and if they do not, then it can lead to an autoimmune disease. Many cells containing antigens and our lymphocytes attack our cells, and if they do not attack our cells, it can lead to an autoimmune disease. I'm trying to figure that one out. So if we have, what you're saying is we have our cells contain the antigens, yes they do, And if we don't attack ourselves, then we don't recognize. So I'm guessing what you're trying to say is the fact that the body has to respond and recognize self cells as being self cells. And if they don't recognize them, then what they do is they will then start to attack our self cells. So it's important that our immune system recognizes what self cells are and what the non-self cells are, in a sense, so it knows what it's attacking and what it's not. But yeah, it is fascinating, and the more you go in, the more you study it, the more you'll learn and get to a point where you're like fully engrossed and become expert in what you're going to study. So it may be that immunology is what you're interested in. It may be that you're interested in something else. It really depends. So like zoology, ecology, it really just isn't. I think my teacher was saying something along the lines of, if we don't get rid of the lymphocytes that kill our cells, then we... right, okay, that makes more sense. Okay, yes. So, okay. Yes, if we don't get rid of the lymphocytes that don't recognize our self cells, yeah, that would make more sense. What they're going to do is the lymphocytes that are going to not recognize our cells as being self cells, they will then start to destroy the cells. And so what you want to do is you want to reduce the number that are going to attack your own cells, because obviously you don't want that. And there are special cells, they're called regulatory cells, that we don't really talk about. They used to be in the OCR spec, but they've removed them out of it. I guess they just decided they're not really wanting you to go into that amount of depth in it. Obviously, if you go on later on and learn, there's so many things, like you'll learn in GCSE, this is biology, and then you get to A-level and you're like, you lied, that wasn't biology, that was like, wasn't even touching the surface of it. Then you get from A-level up to uni and you're like, what? You told me like that and it's nothing compared to what I'm learning now. And you're like, well, yeah, we're slowly giving you the information to build on it so that you can then not become too overwhelmed and also go and find where your passions lie. But yeah, it's so fascinating. Yeah, we do. You're absolutely right. We have so many, and it's about the specificity of the lymphocytes and what they recognize and what they actually are going to replicate into. So if you think about it, clonal selection is important, because clonal selection is about selecting the right lymphocyte that has the antibody against the pathogen. And I had a funny one because I didn't realize, it's a bit of a weird one. So all my life, my favorite meal has been macaroni and cheese. It's like my best meal in the whole entire world was cheese. I love cheese. Macaroni and cheese was just like, give it to me 24/7. I would eat it non-stop. I love macaroni and cheese. And then I had my daughters, and I had my first daughter, and when she was, oh, what age was she? I want to say she was 10 months old. She started getting really bad skin reactions, and so when I took her to the doctors, we thought it was ringworm, but it wasn't ringworm. They were like, she's got a dairy allergy. And so my partner, he's got allergies to proteins, she had allergies, so, and I was pregnant at the time with my second child, and they said, well, she's more than likely genetically going to have the same issues with these proteins, so whilst you are pregnant and whilst you are breastfeeding, what you should do is cut out the milk products in your diet. So I went from loving and having cheese all the time and then cutting it completely out my diet. And I went through all of my pregnancy, my second pregnancy, I went through all of the period of breastfeeding of having no cheese. And I got to the point where my youngest was at a point that I was going to stop breastfeeding. And I was like, yes, give me that cheeseburger, give me macaroni and cheese, give me what I want now. And I had a steak and honour. So in Cambridge we have this place called Steak and Honour. I had a steak and honour cheeseburger, and I was super excited. I was like frothing at the mouth. I was so excited about having this cheeseburger. And the minute I had the cheeseburger, I was so sick and so ill. And what had actually happened is, because I'd cut it out, I'd actually got an intolerance to the milk proteins myself, and I'd actually had it my whole life, but I never knew I had it, because the more exposure you have to it and the more you eat the products, the less of a reaction you get, unless obviously it's anaphylactic, and that's completely different scenario. But because it was giving me just gut problems, I didn't think anything of it. However, because I completely cut it out, those gut problems were extreme. And so that was a really interesting one, where it was like one of those things of, I've never experienced that sort of thing. So then I'm an all or nothing kind of thing as well. So I completely cut it out from my diet. And then my partner decided one day, he's like, oh, I'm going to get us lunch. And we were like, yeah, we're going to have a really nice lunch together. And he ordered a curry, and I was like, oh my God, this is amazing. So if you've ever had a dashum, the chicken ribby, amazing. Dashum chicken ribby, cannot fault it. And he ordered this chicken ribby, and I was like, oh my God, this is amazing. You found like the best tasting curry. I cannot believe you found one that's dairy free, that's this good. And he was like, oh no, it's not dairy free. I was like, what do you mean it's not dairy free? Because he'd been bringing dairy back into his diet and slowly exposing himself. He's like, I didn't care. I thought we'd be fine. I couldn't pick up my daughter from preschool because I was so ill again. However, Dashum is that good. I take tablets to make sure I can enjoy it without having the same experience. And I've been exposing myself more and more. So therefore, it's not as bad. But it's really fascinating how your immune system actually works, and what ends up becoming the majority of your cells that are going to fight or repair, etc. Bit of a random detour, but there you go. There's a personal story for you, how your immune system can attack your own cells and can actually be quietened down, so you're not having as much of a severe reaction. It's why when they say, like with children, their first exposure to things like peanut butter or peanuts, if you're trying to figure out if they're allergic to it, by having the first time you get exposure to it, you don't have a big reaction. It's the second time, because you've already built up a response ready for it, that can then cause more problems. Anyway, I'm coming back to the question you did ask me about the structure. So if we look at an antibody, you should know that an antibody is made up of 4 polypeptide chains. I'm going to draw this very basic right now. So I've got 4 polypeptide chains. Ah, thank you, Slaymays. A star, woo! So exciting. I absolutely love hearing from you guys. Thank you. And it's not just about subscribing. Please do subscribe to my channel. For those of you who haven't already done so. But what's important is it's about the work that you do. I just try and facilitate it. That's all I do. Okay, so we've got number 1, number 2, number 3, number 4. We've got 4 polypeptide chains. So the fact that we've got 4 polypeptide chains tells us straight away that this is a quaternary structure. So we've got 4 polypeptide chains that are held together. Now they are being held together by disulfide bridges. So the fact that they are, yes, this is the antibody. They are being held together by disulfide bridges. So we know that the disulfide bridges means that there has to be cysteines in these locations. Now, what students tend to forget, what they don't think about, is the fact that if I were drawing it very basic, my primary structure is my sequence of amino acids, my secondary structure, my alpha helix or my beta pleated sheet, and then my tertiary structure is where it folds and bends again. So when we talk about the antibody structure, we know it's a immunoglobulin, which means it is a globular protein. A globular protein which has a quaternary structure. The fact that it has a globular protein means that it had a tertiary structure that gave it a globular shape, and then the 4 subunits being held together is what makes it have a quaternary structure. So it's important that you're linking your protein structure together with your immune system knowledge. So we have a globular protein, globular, which means it is soluble. Well, it has to be because it's going to be released through the process of exocytosis from my B cells. Why is it a globular protein? It's a globular protein because it has the tertiary structure and the fact that it is soluble in the blood plasma, because the exocytosis, the B plasma cells that are going to release the antibodies, the antibody is released into the bloodstream, and the bloodstream is obviously made up of plasma, which is basically water with dissolved substances. So the fact that it has to dissolve in the plasma means it needs to be soluble. Because it needs to be soluble, it's therefore a globular protein which has hydrophilic, I always put 2 L's and I shouldn't do that, hydrophilic amino acids. Any hydrophobic amino acids, they will actually be folded in the inside. And that's the other thing to remember, that when you're looking at, let's say we looked at a random protein here, on the outside of the structure, if it's folded like this, this will be filled with hydrophilic amino acids. The hydrophobic amino acids will be somewhere inside, hidden away from the fluid on the outside. Because they are hydrophobic, they will be orientated inside, which makes it fold and bend the way that it's folded. So it doesn't mean that the antibody doesn't have hydrophobic amino acids. It just means that they're folded away from the outside that would be exposed to the water. Okay, so now we need to identify some key parts to it. One of the important parts is this region, and this region here is what's known as the antigen binding site. So it's the antigen binding site, and the reason it's the antigen binding site, my writing is so bad. Binding site. It's funny, I've spent some pens that when I try and write with them, my writing gets worse than with others. Oh no, Unit 6, what's going on? Which one are you doing? Which one are you doing on Unit 6? Oh, bless you. The whole Unit 6 videos are on my channel, so do check out my videos if you haven't already. If you're just coming across me for the first time, every single video that I have out there covers the whole 2-year course for OCR A. It's Biology with Christine, so I've called it the same as what it is here, and it's specifically in playlists linked to your specification. So if it's 6.1.1 cellular control, right, okay, hold on, you're doing AQA. Hold on, let me just, I've got AQA's thing up. So hold on, let me just have a quick look, because it is all interlinked, it's just about where it lies. Is this National 5? It's actually A-level. It's not GCSE. My GCSE video that I did earlier is on the other side. Right, okay, so let me just say, it's just so much Unit 6. You're very welcome, Faith. So phylogenetic tree. So on your specification, hi Tilds, I see you there. So yeah, it's very much similar. It's just you got to find where it links. So the stuff that is about genetic diversity. I'm just having a quick scan. Gene technology, that's manipulating genome and OCR. Hold on, module 6 is... Oh, no, that's responding to internal... Right, okay. That stuff then... Right, okay, so that stuff is 5.1.5, animal response and plant response. In the brain, yeah, 5.1.1. Tills, I'll come back to the agglutinations in 1 second. Control of the heart rate, 5.1.5. Nervous communication, 5.1.3. Synapse transmission, 5.1.3. 5.1.5, skeletal muscle is animal response. Homeostasis is 5.1.1. So you want module 5. Yep, module 5. So basically, whatever your name is, 3 dots, you want module 5 for majority of module 5 for all your module 6 content. Fatima, thank you, Fatima. Sorry, I hate when I don't know who people are. Right, Tills, coming back to you. You want to know about agglutination. So when you talk about agglutination, basically it's about clumping the pathogens together. So give me a second. Let me just put up a picture to show you. So you don't need to know about opsonins as the name for AQA. You don't need to know them as opsonins. You just need to know that they help with phagocytosis. But for OCR, they do need to know that name. So there is my antibody. So if I just bring this all down. So I was just drawing my diagram a minute ago. Let me just move that down slightly. There we go. So there we have our antigen binding site. We've got our 4 polypeptide chains. We've got what's known as our variable region and our constant region. And we also have the role in which they play. So antitoxins for neutralizing, agglutination. So as you can see here, there are 3 of the pathogens all stuck together. And that's because if I call this, there's number 1, there's number 2, there's number 3, there's number 4, there's number 5, there's number 6. There are 6 antibodies sticking. So number 5 is stuck to, let's call that A, B, and C. So number 5 is sticking A and B together. Number 6 is sticking B and C together. Number 3 is also sticking B and C together. And the fact that they've got hinges, what that means is that they can actually bend and move. So if I'm attached over here to one of the pathogens, I could, because of the hinge region, actually have this one attached to a different one. So it's a way of clumping them together. And by clumping them together, it makes it much easier for the phagocytes to come along and engulf and digest them. So if you're OCRA, you need to know that the tagging, so at the bottom here, this receptor binding site, this is where, if they're attached, they are then opsonins, because they are going to make the pathogen more recognizable to the phagocyte for that engulf and digesting. So it's a case of they can either be for neutralization, they can be for agglutination or opsonization. It just depends which one they want it to be for, excuse me, the question that they've asked. Has that helped? You are very, very welcome. Now, I haven't done stuff on the brain in a while. Fatima, are you still in here? If you are still in here, let me quickly show you. Oh, good. Do you know what is one of the topics that most students struggle with? And they have to go over again and again and again. So I would advise to keep going back over it, and hopefully that will make you feel a lot more confident the more you do. No, no, that's not what I wanted to do. What I want to do, where are you? I love the animal response topic area. I love most of biology, to be honest, but it's tough. Don't think. Just to be clear, guys, don't think that teachers don't have to work for it as well. We have to recap the stuff that we're teaching you. If we haven't done it for a while, we have to go back over it again and again and again. Okay, and sometimes we don't have the time to go back over it. Do year 12s normally have a mock at the end of the year? Yes, they do. I'd be surprised if your teachers don't do the mock. Depending on your school, depends on how they treat that mock. So some schools will treat the end of year 12 mocks as being a way of saying whether or not you can continue into year 13. Because if you're not passing year 12, to jump into year 13 without having done any intervention or any work is quite a challenge. And so they will do a mock to show you where you're at, then allow you this summer to try and figure things out. And some schools may even tell you you can't continue into year 13 if you don't hit a grade, as the main mock will be in January ahead of the real A level. Yeah, and it's a good way of doing it. So your year 12 mocks, depending on your school, your year 12 mocks will actually work as potentially being starting predictors for your UCAS grades. So those mocks do matter, and so many students are like, oh, but it's just year 12. It's not. One, it's an identifying feature of where you're at right now, and two, it's going to help your teachers gauge where you're going to be, whether or not you need intervention, what intervention you should be doing. So you yourself should be putting in the work that you need to be successful. Your school, your teachers should be helping you as well. So in my Directorate in Progress role, that was something I used to do. She's lying, I got a U in year 12 and got an A star in the real exam. UCAS don't care. I never said it was for UCAS. You didn't listen to me. I'm not lying. I'm telling you what that grade would mean for the teachers. So just be... No, I didn't. You didn't actually listen to everything that I said. I said the school that you're in will use that as a way of predicting you for UCAS. Your teachers, not what UCAS will accept or not. I didn't say they affect your UCAS grade. I said they affect the prediction that your teachers would do for your UCAS grade. I don't know why I'm arguing with you, actually. That is one of the things that your teacher will use. So what I'm saying is they are a way of you identifying where you're at right now. Depending on your school, depends on what they'll do with it. So I'm not saying it's the right thing for them to do, because some students just basically don't put any effort in, spend the summer recapping, refreshing, and as you've said, you then worked yourself to get that A star. And that's what's important, what you actually get at the end, because that's the grade that determines whether you get into your place or not. But what I would say is, for those students who leave it to the last minute, they're making things much more difficult for themselves when you get to year 13. It is A level, yes, it is A level that we are doing right now. But yeah, it's definitely important to use your mocks as a guide of where you're at right now. Oh, nice. I'm glad you'll be back next year. I have got videos on GCSE if you're looking at GCSE. So if you check out my YouTube channel, you will see there are, if you do AQA anyway, there are some videos for AQA, and I've been doing some lives to help GCSE students as well. But yeah, going back to the mocks, it is a good indicator of where you're at right now and what you need to do. That's what's important. Is checking out your current situation, your current understanding, your current level of written ability as well. Oh, nice. Oh, I hope it comes in as you want it to be. Chills, that's really cool. It's really hard when you've done a test and you're sitting there waiting to get the results back. And there will be times when you look at it and you get a bit frustrated because you'll made mistakes that in hindsight you look at it and you're like, why did I make that mistake? And that's okay because you use that as your guide. So the next time you get a question similar, you don't make the same mistakes. And again, that's why tests and mocks are there. I kind of switch between AQA and OCR. My resources are built for OCR, but I tutor a lot of AQA students as well. So what I do is I kind of jump between this is OCR, this is AQA. But I would say make sure you're looking at your specific specification. Was it the exam contained bits that we hadn't? Do you know, it is so many times that happens. I did that when I would set my students test and they're like, I haven't done that yet, we haven't covered that yet. And it's because as teachers we only have a bank of questions that we can use to build and also to give you real experience. We can make them up, but we don't have the mark schemes like the exam board would for you. So it's better to use exams and past exam questions that have been used in real exams. And so, yeah, sometimes you get these questions where you're like, they're good because you will actually get exposure to stuff you've never come across before. So in your paper two, because you're AQA, in your paper two, when you get to the last question, you will get in paper two a comprehensions part and where you'll have to read some information and then apply your knowledge to it. So that's important for that. And it was to taking in cellulose for making the cell plate to divide plants. Ah, that's lovely. That's a really nice one. That actually it would be more application. When you say it was taking in cellulose, do you mean it was taking in cellulose? Or do you mean that it was producing cellulose and then the Golgi vesicles were moving to the metaphase plate and then fusing together in the building with the enzymes? So you're talking about the cytokinesis part. It was like a diagram of a channel protein in the membrane and cellulose being taken in. Oh, interesting. Oh, hold on. Yes, no, yes, yes, yes. So what it's saying is the cellulose is going into be added together to make, yeah. So that question is still assessing you. So that's that's just such an interesting way of asking the question. That question is asking you about, do you understand that cellulose is a polysaccharide? Because it's a polysaccharide, it's too big to get across the membrane, so therefore would have to go through a channel protein because of its size and because it needs to get across the membrane to get to the area where the wall would be built. And where did the cellulose come from? The cellulose came from, yeah, that's what it is. It's so cool. It is, everything is linking it together. So it's about that gripping of it. But what I'm finding really fascinating is because they're making it out that the cellulose is going through the channel. The cellulose would be being made in the smooth endoplazmic reticulum and then moving in the vesicles. See, now you've got me intrigued by it. Give me a second. I'll have a look and see if I can. Oh, excuse me. Yeah, so I think that's really interesting because the cellulose doesn't move across at all, which is why it's really fascinating what they've actually said. Um, I wonder if it's in. Hold on, let me have a look at the bank of questions I've got and see if I can find it. Um, I need to have a look. But this is where some exam boards are. They come up with these questions, and then it's like you have to think about. Oh, how did it go, Daryl? I'll definitely take a bit. Yeah, please do. Send it to me via email. Oh, I'm really pleased. Did you find the nucleic acid nucleotide? The notes clutched up. Oh, this is where I'm not down with the lingo. You need to help me out here. What is clutching? So my email is either ukbiotutor@gmail.com. So my web address. Oh, save me. I love that. Or you can send it to christina@aiqchat.com. Either of the two are my email addresses. I love that. Is this a new one though? Clutched up. I'm trying to think how and where that would have come from. Like a clutched bag that saved you. I don't know. But I'm pleased that they've helped. I'm really pleased. I'm glad that you're finding them useful. The only question was a 2 marker about some random formula for DNA replication and bases. Oh, interesting. A random formula. Oh, hold on. Was this the G? Darren reminds me, are you AQA or OCR? No idea what that meant, but otherwise it went well. Is it the one where they give you like C plus something or other OCR A? Okay. I was saying that was an AQA question, that one, where they gave you these are the DNA bases, the number of bases, and C equals G plus blah, 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 and you had to work it out. It was a lovely question, but it was an interesting one. But it was AQA, not OCR. So unless your teachers used an AQA question. Hold on. Let me see if I can find that one that we're talking about. Because I actually did it with one of my students the other day there. And I did like it. Yeah, try and find it for me if I can't find it. I think I've just guy. Hold on, if it's an OCR one, I can access that one a lot easier. Hold on. Let me see. So, I mean, you're not talking about mission style, are you? She was saying it was DNA replication. I didn't think it was that one. Six different bases. Strand one, strand two. And percentages. Filling the table. Oh, how interesting. A fill in the table. I think they might have used an old style question on that one then. Yeah, I'm not sure. I'm trying to find it. I think they've used an older question. It would be interesting to know. Oh, hold on. Oh, maybe that's too easy. Maybe that one. Just soften that. I'm not sure what it is. See you on Wednesday and I'll be sure. Oh, thank you. Tils, have a good one. I am actually going to finish now myself because I can hear my voice is starting to go and I've got some sessions for. Yeah, I don't think it is there. I think they've used an old style question. But I'm pretty sure it was OCR because you can have them. No, we can tweak them. We can tweak them. It's although you think it's like one, it doesn't necessarily mean that it is that one. I use different examples all the time because I find that they're a really nice way of bringing it through and assessing students on questions they've never seen before. So I do like to bring in some random ones sometimes. Right, guys, I am going to go. Thank you for joining me. I will be back on. When will I be back? Wednesday at 9 o'clock. If you want to join again, if you have questions, please do ask away, send me emails. I will get a list of stuff and I will add it to the repertoire of what we do. But thank you for joining me. Have a good evening and keep going. It's hard, it's tough, but you can and will be successful if you just keep going. Little and often is what's important. But thanks for joining me, guys. Bye.